are many different kinds of viruses, yet all viruses are characterized by the fact that they have nucleic acid towards their middle inside and have a protein overcoat around their exterior. Now, viruses are inert and inactive as long as they are outside living cells. But once they're placed inside specific living cells, they spring into action and can multiply many times over. A single virus particle, for example, can produce thousands of progeny within a half an hour after infection of a given cell. Now, there are three general classes of viruses: those that affect bacteria or germs, viruses that infect plants of various kinds, and viruses that affect animal cells. Here, for example, is an example of the latter, a model of poliomyelitis virus, magnified about 65 million times. It's the animal viruses and similar systems that we'll be talking about today. Now, if you take a crystal of poliomyelitis virus made up of many thousands of individual particles and look at it by means of the electron microscope, this is what you will see. Every one of those little round circles that you see there is a molecule of poliomyelitis virus. Now, poliomyelitis virus has been known for a long time because it seems to have caused disease in Egypt. Here, for example, is a hieroglyphic showing a man with a shriveled up leg, presumably due to the fact that he may have had poliomyelitis. Still more recently, viruses have caused still other diseases. A virus actually is responsible for the greatest outbreak of disease within the history of mankind. And I refer, of course, to the widespread outbreak of influenza in 1918. During that winter, 500 million people had influenza, and of these, about 20 million died. We lost here in the United States about 400,000 people during that winter. For example, in San Francisco, there are big ads placed in the newspaper advising people to wear masks to protect themselves against influenza. And in the South, newspaper articles carried indications that people were advised to stay away from the churches and shows as a means of protecting themselves against this disease. Now, it's the diseases that are caused by viruses that are going to interest us today. We'll learn a bit about these virus diseases and about similar test systems. And for this, I'd like to introduce to you my colleague, Dr. Harry Rubin.
We think of virology as the study of the physical and the chemical nature of viruses. Then its origin is rather easy to date. The most commonly accepted date is 1892, when Ivanovsky discovered that if we extracted material from a mosaic disease in tobacco, we could filter this material through a filter which would hold back the smallest bacterium, and yet what passed through onto the other side was capable of causing the same disease when inoculated into tobacco plants. However, if we're more liberal and we consider in virology the study of virus diseases, then its history is far more venerable, dating way back before the Christian era. Perhaps the best studied and the best documented of all the virus diseases in history is the scourge of smallpox, chiefly because of its widespread nature and the severity of the disease. It was known before the Christian era, particularly in Asia, and was brought into Europe in about the 16th century. There it caused some widespread scourges. Through all the history of smallpox, the ancients, as well as more modern people, were quite superstitious about its nature. But remarkably enough, they made some quite astute observations. They realized that it was a single disease. Furthermore, they realized that it was contagious, that it spread from one human to the next, and they even had a pretty good idea of how it spread. They were aware of the fact that the scabs, which were the lesions of smallpox, contained some kind of material which, when inoculated into a normal human being, caused the same kind of disease. And further, and perhaps even more remarkably, they realized that when a person recovered from smallpox, that he was immune, that he could not be infected again, and there were further epidemics of the disease. Now, some unsung ancient put two and two together, probably in China, and realized that if he took the material from the smallpox scabs and deliberately inoculated a human being, he could cause the disease. If this was done under the proper and optimal conditions, the individual would develop a relatively mild disease and would be immune to the more severe outbreaks of smallpox. The only unfortunate aspect of this practice, which became very common in China and in Turkey and later in England, was the fact that a large number of people died because it could not be very well controlled. However, this practice of inoculation, which was called variolation, was the logical predecessor to what became the most monumental discovery in the history of virus diseases, if we measure such discoveries by their importance to the survival of human beings. This was the discovery of the present-day technique of vaccination against smallpox. It was believed in the rural areas of England that if a person contracted a disease from cattle, which was known as cowpox, that this person, when he developed a mild disease himself, somewhat like smallpox but much milder, was thereafter immune to the disease. In general, this belief was scoffed at by the men of science, but there was one, Dr. Edward Jenner, who did not scoff at it, who believed that it was true, and furthermore did some experiments. He removed scabs from cattle which were suffering from cowpox and inoculated this material deliberately into human beings, beginning with his own son. He found that these human beings would contract a mild case of cowpox and were thereafter immune to the more severe disease of smallpox. This process of vaccination, called vaccination for vacca, which is the Latin word for cow, this process of vaccination became commonly adopted throughout England, but not generally very popular, as indicated by this cartoon of the time. Many people believed that when they were inoculated with the material obtained from cowpox, that they themselves developed partly into a bovine nature. It's important to realize that these developments in vaccination occurred something like 100 years before the discovery of the true nature of viruses. Of course, we've learned a great deal about viruses since that time, but there's been very little in the way of a basic improvement on Jenner's vaccine since that time. However, we must realize that before Jenner could even think about such things as vaccination, he had to have the accumulated knowledge of many centuries, the basic information about the nature of the disease and the spread of the disease, and immunity in smallpox. Today, we're in the process of building, in a much more concentrated and systematic way, further knowledge about what viruses do to animals and what they do in particular to cells. The basic tool of the virologist who is interested in the interrelationships between viruses and cells, the basic tools are represented here on the table. The first, one of the most important, is a pipette to obtain precise measurement of quantities. Secondly, we have fertile eggs, which are the source of embryos, illustrated here, where we have 2 embryos. And finally, we have a petri dish, a simple laboratory vessel. The pipette is for accurate measurement, as I've said, and without precise techniques for measuring fluid volumes, we cannot carry out accurate scientific work. The egg itself has a long and honorable history in the study of animal viruses, and in particular, it has an honorable history during the era when it was necessary to study the multiplication of viruses in whole organisms. Because of all the whole animals that it was possible to work with, the embryonated egg, the fertile egg, was the simplest of all these. The petri dish symbolizes one of the important new developments in animal virology. Beginning about 10 years ago, techniques were found for removing embryos from the fertile egg, dispersing the cells, and placing them in tissue culture. This is done in the following manner. We simply crack open an embryo, remove some of the membranes, remove the living embryo, place it in a petri dish, then remove some pieces from the embryo, and place the embryo in this mixer. The purpose of the mixer is to agitate the cells from the embryo and put them into a suspension, which we can then later handle. What we do when this is finally completed and the cells are in suspension, we can place the cells in a petri dish where they will settle on the glass, stick there, and multiply. If now we're interested in studying how much virus we have in a particular preparation, we simply take the virus preparation, measure very accurately the amount of fluid that we're taking up, and then add it to the cells. Within a period of several days, the virus multiplies in the cells and kills the cells, and by counting the areas of dead cells, we can determine how much virus we had to start with. Now, the developments in tissue culture have had importance for more reasons than one. The first and the most obvious is that it now becomes possible to study the multiplication of virus under conditions where we can continuously observe the infected cell, from the moment the virus gets into the cell until it destroys the cell. Secondly, in addition to continuous observation, we can make very precise measurements of how much virus was present in a particular suspension at any given time. And finally, it's been possible to use cells from human beings for studying the virus, the multiplication of myelitis. A particularly popular tool has been the HeLa cell, derived from a human cancer. And this supports very beautifully the growth of poliomyelitis virus. The events which follow after poliomyelitis virus is added to human cells in tissue culture are illustrated here. A single human cell growing in tissue culture. The black dot represents a single poliomyelitis virus particle which adsorbs to the cell and penetrates. For several hours thereafter, we see no change in the cell whatsoever, but there are changes in the virus particle. The first thing we do see is a change in the center of the cell, the nucleus, followed by virus multiplication in the cytoplasm, and a change in the shape of the cell. Finally, the cell bursts, releasing many virus particles, which can then reinfect the surrounding cells. This process of the penetration of the virus, the multiplication of the virus, and its release, followed by the death of the cell, is repeated over and over and over again, each cell releasing hundreds and even thousands of virus particles to infect the surrounding cells. And after this cycle has been repeated many times, we have an area in the tissue culture of dead cells. This area is what is known as a plaque, or an area of dead cells among a group of living cells. By simply counting the number of such areas of dead cells, we can determine how much virus we have put into our tissue culture, and we therefore have an accurate way of measuring the number of virus particles that have been present. And by studying the changes in the cells, the time of release of virus, we can get a good idea of how a single virus particle can get into a cell, multiply in that cell, and destroy the cell, and consequently destroy a group of cells. What does this mean, however, in terms of virus disease? here we've seen what a virus does to an individual cell or a group of cells. Can we project this picture into the human organism? In the following film, we'll illustrate the more complicated sets of events which occur when a virus particle and a group of virus particles infect a human being and multiply in tissues of various types and finally cause the typical disease of poliomyelitis. Here we see what happens when poliomyelitis virus infects a human being. Infection is thought to take place through the gastrointestinal tract, resulting in infection of cells which line the walls of the intestine. Probably these are lymphatic cells. The virus multiplies there and spreads through the lymphatic system into the lymph nodes. Very frequently the infection is stopped here, but occasionally the virus multiplies past these filters, the lymph nodes, and when it does, it gets into the bloodstream, where it's carried to all parts of the body. A critical place in the body, however, where the virus begins to multiply is the central nervous system, depicted here, where we see the spinal cord. Multiplication of virus in the central nervous system, the spinal cord and the brainstem, is what produces the disease which we commonly know as poliomyelitis. When the virus multiplies in cells of the central nervous system, it reproduces the sequence of events that we saw in the multiplication of the virus in tissue culture. However, this time we're dealing with neurons, the essential cells of the central nervous system, the cells which control the movement of muscles, or in the case of the brainstem, the cells which control such important functions as breathing. If the virus multiplies in enough of these cells and destroys their function, we get paralysis of a limb, in case the neurons are those which supply a limb, or in the case of the brainstem, we may get paralysis of those mechanisms which are responsible for breathing, and hence we have to employ such devices as iron lungs in the therapy of poliomyelitis. This is the illustration of only one disease caused by viruses. There are, of course, a whole multitude of such diseases, most of which are commonly known as cytocidal viruses, or viruses which kill cells. This includes yellow fever, encephalitis, rabies, smallpox. But there is another class of viruses which is not so well known to most people, but perhaps in the long run may prove to be considerably more important than the cell-killing viruses. This class of viruses is called the tumor viruses. And these viruses, instead of getting into a cell and multiplying there and killing the cell, multiply much more slowly, and instead of killing the cell, they cause the cell to multiply under conditions where it normally would not multiply. The first of such viruses, which was cleanly established as a cancer virus, is the so-called Rous sarcoma virus. This was discovered by Dr. Peyton Rous in 1910 at the Rockefeller Institute in New York. It was recovered from a cancer called a sarcoma growing in the breast muscle of a Plymouth Rock hen. Dr. Rous was able to reproduce the cancer by making an extract from the tumor and inoculating it into another chicken. Since that time, a great variety of viruses have been found to be associated with cancers, but perhaps the best studied is the Rous sarcoma virus. The sequence of events which occurs after infection of a cell with the Rous sarcoma virus is illustrated in the following film. Here we see a group of normal chicken cells growing in tissue culture. We've introduced the Rous sarcoma virus, again this black dot. This comes in contact with a susceptible cell and disappears into the cell. It begins to multiply there. And we begin to see some changes in the cell. Increased granularity, swelling of the cell. The cell begins to divide. And you'll notice that the normal cells remain quiescent. They've reached the limit of multiplication and they're crowded in the tissue culture. This doesn't seem to bother the infected cell. It continues to multiply in spite of the fact that we have here a confluent sheet of cells. And in this we have one of the more important characteristics of the cancer cell, that it does not recognize the limitations imposed on normal cells. the cancer cell continues to multiply and it continues to produce and release virus. Ultimately, because of the fact that the cells pile up on top of each other, we get a microtumor, a small tumor growing in tissue culture, growing on top of a single layer of normal cells. What you have just seen is an animated film of what happens when Rouse sarcoma virus infects chicken cells. An animated film is nothing more than an idealized version of what we think happens when the virus gets into the cell. To take photographs of what's actually going on is far more difficult, but I want to venture to show you the first film we've ever taken of what happens to normal chicken cells when they're infected with Rouse sarcoma virus. This may take some of you back to the early days of the cinema, to Charlie Chaplin photography, but nevertheless I think it's worthwhile for you to look at to realize that not all is peaches and cream in this business. Here again we see a group of chicken cells growing in tissue culture, only this time it's the real thing. It's perhaps a little bit difficult to distinguish individual cells, but the square is directed on a group of cells which will become transformed, which will become altered by infection with Rouse sarcoma virus. You'll notice that these cells are rounding up. In this still, they've already rounded up, and they're very active cells, in contrast to the cells in the periphery. These cells are now beginning to divide, and divide rather rapidly, caught again in this still. In the meantime, the normal cells in the periphery remain relatively quiescent. There is some type of inflammation which prevents them from multiplying. The Rouse sarcoma cells, the cells infected with Rouse sarcoma virus, continue to be very active and continue to multiply in this fashion. What we have seen is the simplest possible sequence of events which follows the infection of a cell by a tumor virus. We have here none of the complications of the reaction of the whole organism itself. We have a simple system. What happens when we put the virus into the chicken? We believe that a series of events quite similar to what we've seen in tissue culture occurs. We know that when we make infection, within a few days, we can see a tumor, a cancer. By the end of the week, this has grown to considerably large proportions. The tumor spreads throughout the animal and eventually causes its death. We don't know whether all tumors are caused by viruses. However, we feel that in this system, where we study the infection of individual cells in tissue culture with the Rouse sarcoma virus, we have an ideal system for studying the onset of malignancy, and this will have some application in the onset of other malignancies as well. We've seen some of the events that have occurred in modern virology, the improved understanding of what happens when a cell is invaded by a viral parasite. As a result of these modern investigations, we've been able to make a number of important improvements and refinements in vaccines, in methods for preventing virus diseases. We can very quickly isolate a virus from an epidemic. We can identify it. We can make a vaccine against it. In addition, we can grow viruses in tissue culture, which it has not been possible to grow before. As a result, we have such developments as the Salk polio vaccine. However, there has been no fundamental improvement in our methods of dealing with viruses. Vaccination is still the only tool that we have for preventing virus diseases, and strangely enough, we have no way of treating a virus disease once the symptoms are seen. When we consider the reasons for this, it seems quite obvious that this should be so. After all, viruses are made from the same building blocks as cells are made from. And furthermore, and perhaps more important, the virus has no machinery of its own for making more virus particles. When it gets into a cell, It usurps the normal machinery of the cell, which usually goes for making more cells and more cellular components. It usurps this machinery for the multiplication of virus. And as a result, we get damage to the cell of some kind or other. Either we kill the cell or the cell becomes a cancer cell. Therefore, it doesn't take much imagination to realize that any chemical which would interfere with the growth of a virus has a good chance for damaging normal cells in the body. And as a result, we have not yet found anything which could destroy a virus without destroying the host cell. However, I think we can take heart from the obvious fact that human beings during their evolution have learned to deal somehow quite effectively with virus diseases. Almost everyone has recovered from a number of virus diseases. The body handles these diseases with a great deal of dispatch in most cases. I think it's realistic to expect that within the next few years we'll learn a great deal more about how the body deals with virus infection. At the present moment, we know very little about this. We only know that the classical immunological mechanisms, the classical defense mechanisms that we've come to understand in bacterial diseases don't seem to apply very closely in the case of virus diseases. I think that once we've gained some insight into these problems, we may be able to help the body and to help the cell to help itself. In the meantime, we, as virologists, obtain a great deal of satisfaction from the realization that we're learning something about a very complex but fascinating biological problem.
Virus was produced for the National Educational Television and Radio Center by KQED San Francisco, in cooperation with the Virus Laboratory of the University of California at Berkeley. Featured in this program was Dr. Harry Rubin, Professor of Virology. The series was made possible by a grant from the National Science Foundation. This is National Educational Television.